Hi, I'm Dannielle Engle, an assistant professor at the Salk Institute. My lab focuses on pancreatic cancer, which will soon become the second leading cause of cancer-related death in the United States. We try to find new therapeutic strategies using a three-dimensional culture system called organoids, which my team is going to show you.
Hi everyone, my name is Shira and I am a research assistant in Dr. Dannielle Engle's lab here at the Salk Institute. Today, we want to show you how we research pancreatic cancer by using organoids. Organoids are mini organs that grow in a 3D plane. We take the cells from patient biopsies and grow them in a transparent gel called matrigel. They work as avatars for human patient tumors, and we can make them from any organ. In our lab, we choose to make our organoids out of pancreas. We make these gel domes that act as houses for millions of organoids. These organoids have been growing for several days, and they are nice and healthy, so it's time to expand them and give them a bigger home to keep growing. The gel is collected, spun in a centrifuge, and separated so we can just get the organoids. Once we have the organoids separated, we give them new matrigel so they have enough room to keep growing. We add media, which is nutritious and specifically made to encourage the cells to grow. To ensure they are healthy, we look at them under a microscope and see them forming round spherical structures with lots of extensions and connections. They're happy! Just like us, they need food, water, and a lot of attention to thrive. When they are unhappy, they squish together and look dark, indicating we may have to replenish their food or media soon. Our domes overflow with millions of organoids. We grow them on such a large scale so that we can ask big questions and test big ideas.
Hi, my name is Xiaoxue, and I'm a grad student in Dr. Dannielle Engle's lab at Salk. One of the experiments we do is called pharmacotyping. This is closely linked to the idea of precision medicine. Pharmacotyping is used to measure, first, which chemical agents we can use as a therapeutic treatments, and second, what dosage should be used to interfere with the organoids. Because the organoids can be derived from human patients, we can use that to test what drug we can recommend for the patient treatment, and also how much to be given to the patients. During the experiment day, we place our organoids in this tiny plate that contains hundreds of tiny wells. This is what we call the seeding stage. Then we get help from our special assistant, which we call the high-precision digital dispenser. After we set up the experiments, the machine will tell you the steps. First, load the cell plate. Second, add the therapeutic agents. Each plate can screen for 6 to 8 chemical agents, and we can compare and quantify cell actions by counting how many cells survived after the treatment. Third, press enter. It will start moving and adding very little dots to each of the wells. The amount is so small that we cannot do it by hand. We can tell the machine exactly what to do, like making fun patterns. We're only adding a small amount of liquid, but to the microscopic cells, that is a lot. We now come to this exciting step of pharmacotyping, seeing the results. We can measure the numbers of live cells with another assistant, Cell Imager. Our imager will capture how many live cells are, and that will tell us these organoids were responding to which drug and how effective is each drug in killing the pancreatic cancer cells. Higher dosage will harm cells more, so we expect there are less live cells in high dosage wells. This we can compare with evenly distributed conditions right after the seeding stage.